seconds of your attention at the end, so that we all switch our cameras on and we all take a group picture, okay? So thank you very much, and I hope you enjoy it.
So good afternoon, everyone. It's a pleasure to be hosting this session. Thank you, Joanna, for the kind introduction. So it is my great pleasure to introduce Kate Downs. She'll be talking to us about the human genomic revolution. Kate is from the UK, where she got her degree in genetics and master's in molecular genetics. She has afterwards moved to Cambridge, where she has worked for several years at Wellcome Sanger Institute, first as a research assistant before getting her PhD, which she got from the University of Cambridge. Over the years, if you had a chance to look at the publications of Kate, she has several high impact publications, such as in Science and in Nature. And she has been enrolled in many exciting projects regarding human genome projects. More recently, she has been responsible for developing the thrombogenomics. This is a platform to diagnose people with bleeding, thrombotic and platelet disorders, which has been recognized worldwide. And right now, she's a clinical scientist for the NHS. And she works at the Cambridge Genomic Lab, where she's a lead scientist for rare and inherited disease. So, Kate, it's our great pleasure to have you here this afternoon, and the screen is yours.
Thank you. That was a nice introduction. Thank you very much. Okay, let me share my screen. Can everybody see my screen? Yeah, I think so. Mm-hmm. Okay, so yeah, I'm going to give us a whistle-stop tour of what's been happening in genetics for the past 20 years. And I guess also at the same time, what I've been doing for the last 20 years. So I've called this talk a little bit the Genomic Revolution, because I really do feel like we've moved so far in the last few years. And we've got a really exciting future in genomics. So I know some of you guys are geneticists and molecular biologists, but I know that there's some people from lots of different specialities. So I hope that that my talk can speak to all of you. So you can contact me on my email address there, and then I'm also on Twitter on Kate_Sci. So let's get going. So see if I can share my screen. So I guess just before we turn to the 2000s, I wanted to kind of set the scene. And so this is a picture of me. At that time, I had blonde hair, and I was at the University of Liverpool starting my undergraduate degree. And at this time, you know, we really didn't know a huge amount. There was only 6 universities in the UK that were offering genetics. And I looked through the course that I did today when I was writing this talk, and actually we were looking a lot about gene regulation. We knew a lot about genetic variation, but we didn't really know much about human disease. And of course, in 1997, the Human Genome Project was underway, and that was the really big thing that was happening in genetics at the time. So after I finished my degree, oh, sorry, no, I've jumped ahead. So for human disease, as I said, we didn't really know that much. So for common diseases like heart attacks, cardiovascular diseases, stroke, autoimmune diseases, things like this, we knew that there was a heritable component from twin studies. And actually, I'm an identical twin. So this is like a little picture of my identical twin and me. So we knew that identical twins, when you compared them to non-identical twins, they would be more likely to share common diseases. And also, we'd done some studies at this stage where linkage studies had been done in using gene markers, and we'd found already things like the HLA locus was associated with specific autoimmune disorders, but there really wasn't much else known. For thinking about rare disease, well, of course, we've known about rare disease for a long time, even before we actually knew the genetic cause behind those diseases. So this pedigree you can see here is Queen Victoria's pedigree. And in her family, a lot of the males had haemophilia, which is an X-linked blood disorder. So at the turn of the century, 1997, we knew about blood groups and sickle cell disease. We could do genetic testing for cystic fibrosis and haemophilia. We could also see, look at people's chromosomes and see whether or not they had extra copies of different chromosomes. So for example, if you have an extra copy of chromosome 21, then you have Down syndrome. And then I think because of the necessity around specific disorders like breast cancer, we did understand quite a lot about the BRCA1, BRCA2 genes that were associated with inherited cancer. So I guess the theme of my talk from now on is really going to be split into this kind of common disease and what we've learned about this over the last 20 years, and also the monogenic side, the rare diseases. And at the end, I think you can hopefully see how we start to join in the middle. So in 2000, I'd finished my degree and I went to the Sango Institute, but actually that wasn't what was exciting at that time. What was really exciting is that we were finishing the Human Genome Project at the Sango Institute. And that was a huge feat. That was a 13-year project to clone and sequence using Sango sequencing the human genome. I mean, it's an enormous project and it cost a lot of money, and thousands of people were involved in that project. And so I came along just towards the end of that, and actually the work that I got involved in was the work around the variation that we were starting to see in the work that was coming out of the Human Genome Project. So when they sequenced the human genome, they took 14 people's DNA, smashed it up into little bits, cloned that into different types of clones and sequenced them. And when they aligned them, they started to see differences between different clones. And of course, these are what we'd now assume are polymorphisms. And at the time, we had no real idea of how frequent these polymorphisms were. We knew that some of them caused disease like in cystic fibrosis, but in the whole genome, we didn't really have a good idea of what this variation looked like. So these were the projects that I was involved in. So we first had the Snip Consortium project where we were testing 12 Caucasian people to see whether or not a SNP that we'd found in the human genome pileup was actually different in the DNA of 12 different Ceph individuals. Luckily, we decided to expand a bit and start to think about different populations. So by the time we started to do some of this work with the International HapMap project, so haplotype map project, we were looking at, I think, about 250 people's DNA from different populations across the world. And this was really allowing us to see differences in frequencies of this variation in different populations. But it also led to us having a really good understanding of the structure of variation in the genome. So the haplotype map project, so that's quite a specific thing, the HapMap project. So a haplotype is a piece of DNA that's more often by chance passed on from a parent to a child. And this really allowed us to start thinking about what the next step was in our projects. So I'll come on to that in a second. Someone's going to have to tell me when to stop because I've lost my clock. Okay. So, so this HapMap project really started us on the way to being able to do what we now call genome-wide association studies. And the first step of that was to be able to genotype lots of different variation in a person's genome using these genotyping chips. So the first kind of versions, large scale versions of these genotyping chips were about 500,000 SNPs. And all of those SNPs were chosen from the HapMap project and those Snip Consortium projects, where we'd worked out that specific genetic variants could be used to tag or to provide the same information as other genetic variants. So what that meant was we could be quite picky when choosing which variants to put on these gene chips, because we knew we had redundancy in some of that information. So the next stage, well, okay, no, for me, what I did was during that time, so I left my degree at Liverpool, and I didn't do a great job of my degree. I had a lot of fun, but I didn't do a great job. And as I was at the Sango Institute, I was really inspired of what's going on. So at that point, I was really keen to do a PhD in the future. And I knew that because my degree score wasn't great, it would be good for me to do a master's. So I ended up doing a part-time MPhil in biological sciences with the University of Liverpool while I was working at the Sango Institute. And this study was based on the work that I was doing while I was at the Sango. So my hair color's changed because it changes quite a lot. So I've kind of tried to reflect that during this talk. So the next thing that comes is based on the work that we've done with these genotyping chips. So lots of people at this time, this is 2005, were saying to the Wellcome Trust, who is one of our big funding providers for science studies in the UK, lots of people were saying, well, can we do an association study now to compare some patients and some controls, genotype them with these genotyping chips and see if we can find genetic variants in the genome that are associated with specific diseases. And the Wellcome Trust quite cleverly said, well, actually, we've got lots of people asking us to do this. So rather than fund 7 different studies, let's put those studies together, make them use the same controls, and each of those cohorts of diseases have to provide their own patients. So that's what the Wellcome Trust Case Control Consortium was. It was a huge effort, 3,000 shared controls, 7,000 patients from 7 different disease areas, all sequenced, all genotyped with these genotyping chips that we just discussed. And so just to make sure that we all know what a genome-wide association study is, I'll just quickly go through this. So if you take a bunch of controls and a bunch of patients and you genotype them for lots and lots of genetic variants, what you're looking at when you look at your results is you want to find genetic variants that are more frequent in the patients compared to the controls. And that would mean that in patients, if you have that genetic variant, you're more likely to be a patient than a control. So we start to see this variation that's associated with diseases. And these are the results that we published in 2007. So there was 7 disease areas: bipolar disorder, coronary artery disease, Crohn's disease, hypertension, rheumatoid arthritis, type 1, type 2 diabetes. And these plots are what we call Manhattan plots. And this one's a bit squished, but you can see the green bits, I hope. And actually, these green bits are the p-values of genetic variants. Each dot represents a genetic variant in the genome. And these the green ones are the regions of the genome which have genetic variants that are associated with a disease. So you can see there's not a lot of green dots. In fact, for bipolar disorder, they didn't actually find anything that was of statistical significance. For type 1 diabetes, you can see we've got these region which is shared with rheumatoid arthritis, and this was the HLA region that we'd already known about. But for type 1 diabetes, you can also see a little region here, and one there, and one there, and one there. And so after this study, I'd got to know all of the PIs that were associated with these diseases, and the type 1 diabetic lead asked me to go and join his lab. So I did, and I focused on type 1 diabetes in the University of Cambridge. So the first things that we were doing was replicating these associations, and we published some of that work in 2007. But I just wanted to kind of flip to what was happening in the rest of the world at this time. So of course, we were just talking then about genotyping chips and genotyping 500,000 genotypes in people. But at this same time, there was a whole different sequencing project going on in the world. So this is 2007, and there was new methods now available to do whole genome sequencing. So remember the Human Genome Project took 13 years to do, and there was now methods that could probably do genome sequencing in about 3 months. So there was 2 big projects going on. One by a pharmaceutical company owned by this guy called Craig Venter, and 2, a public funded sequencing project, which was aiming to sequence James Watson. And this really was a war of projects to try and to make sequencing accessible to the general research community, and it not be used by just pharmaceutical companies. So actually both of these genomes of these 2 men was published in around 2007, but I think it really opened the way for whole genome sequencing to be used within academia as well as within pharmaceutical companies in the future, in the following years. So for me, I'd moved to the type 1 diabetes labs in Cambridge, where we'd continue to do some genome-wide association studies, and these were getting bigger and bigger. We were also doing some cell function work, because of course, if you find a genetic variant that is associated with a disease, you don't know what that genetic variant is doing. It's likely not sitting in a gene. It's likely sitting somewhere between 2 genes. And so some of these cell function studies, these expression studies, and then these studies looking at gene regulation really started to help us pinpoint where these genome-wide association signals were actually having a mechanism in terms of how they were causing disease. So also at this point, the genome-wide association studies were getting bigger. So of course, in 2007, we published with 1,000 cases and 3,000 controls. And we were starting to, do 5,000 cases, 5,000 controls due to studies that had been biobanking patient data for years. So we were starting to find more and more genome-wide association signals. So bits of the genome that were associated with disease. And at this point, so I was at the University of Cambridge, and I was in the type 1 diabetes lab, and I did my PhD and finished it in 2010. So my thesis was the genotype-phenotypes associations in type 1 diabetes. So it was exactly what was going on at the time. So of course I was working on type 1 diabetes, but my colleagues across the world were also doing very similar projects in other types of autoimmune diseases, but also in all kinds of common complex diseases. So just to make sure that we're kind of all on the same page here, this is where I'm going to introduce you to one of my favorite graphs. This is our allele frequency effect size graph. So this is just to try and explain where the genetics of common variation, so that's this bottom corner, these are the variants with high allele frequencies that we find during GWAS studies, but they have a really low effect size. So of course, if you had one variant that makes you more likely to have type 1 diabetes, it doesn't guarantee that you're going to have type 1 diabetes. It just means that you're tipping a balance in your immune system. And that's something that's very different compared to the rare variants that we see in monogenic disease. So these have very low allele frequencies. They're very rare, but they have really high effect sizes. And this is what we were talking about before when we were thinking about cystic fibrosis and some of those other rare diseases that we'll come back to later. So this is where we're in 2010. And through next generation sequencing, so these genome-wide sequencing methods, we were starting to explore more and more genes associated with specific diseases, but these studies were still really quite expensive. So you'd have to take a group of patients with a really specific disorder to do some sequencing in the right genes to then find the reason why they had their disease. And then these genome-wide association studies, as I said, were getting really big. So the next step for me, probably my hair was changing again. So the next step for me was to think about postdocs. And I was lucky enough to just move across the road from the building that I was in working on type 1 diabetes into a hematology lab. And I took a little bit of a sidestep and was involved in this 5-year project called the Blueprint project. And it's a little bit of a sidestep, but everything we did in that project, we're still using that when we're thinking about mapping gene regulation in common genes. So this was about 2011, and this project aimed to map 32 blood cell types for the regulatory regions. So we were doing ChIP-seq to look at histone modifications. We were looking at transcription factor binding in these cell types. We were doing RNA sequencing to look at which genes were switched on and off. And all of these projects were really useful when we were thinking about those genome-wide association study hits. So, of course, we were taking these genetic variants that we'd identified, and we were starting to really pinpoint which gene and which cell type these variants were involved in. We were starting to be able to think how these variants altered protein function and how they altered cellular pathways, and really from thinking about cellular pathways, how we could potentially treat or use specific treatments prophylactically for these diseases. So at the same time, we're moving a bit forward. So I did have blue hair for a little while. It didn't last very long. I think it was quite bright. So in the same department, the hematology department, we were doing this epigenetic work, but we were, of course, in the Department of Hematology. So we were mainly focusing on cardiovascular disease. And this is about 2015. And I think probably at that time, that's when Joanna came to join me. So Joanna, who's one of our hosts today, she came to join me to work in my platelet function lab. And so we were doing platelet function work. We were looking at platelet gene regulation, and we were doing larger and larger genome-wide association studies associated with stroke risk, with cardiovascular disease risk, and also looking at platelet function. So back to our graph around common variants with small effect sizes versus rare variants with large effect sizes. Well, we started in 2015 to be able to see results coming from huge studies. So the UK Biobank study had got 200,000 people by 2015, where they'd done all those genotyping chips in those people. And they also had phenotypes like platelet count, platelet volume, red blood cell count, hemoglobin. So we could really start to learn a lot about platelets in those individuals. So this is where we start to be able to move up and between those two ends of the spectrum. So we started to see these low frequency variants with medium effect sizes. And that really started to get exciting because we could start to think about how this was an actual spectrum rather than two very distinct disease mechanisms at the end of the spectrum. So in hematology, we were also thinking about applying what we knew from these platelet studies in lots of thousands and thousands of people into, well, actually, what about the people who have diseases of platelet disorders? So what about the people who are at the very ends of the traits, the people who have hardly any platelets who bleed a lot because they have platelet disorders. And there were also disorders where people have lots and lots of platelets, and that can cause them to clot. So can we start looking at those end traits? So at this point, we could start doing we started to do this thrombogenomics project. So this was where we could target 100 genes using a sequencing panel. And we were running this as a clinical diagnostic service. So of course, we were in the university. We weren't an accredited laboratory, but we were providing a service that was just as good, and if not better, than some of the services that were available in the NHS. And what we were doing, we were running these projects as partners with our NHS lab. So if we found genetic variants that we thought were causing somebody's rare disease, then we were checking those genetic variants in the accredited lab before we sent out a report to the patient. So at the same time as doing clinical diagnostics, in 2015, we were also starting to think about how we could do whole genome sequencing for gene discovery. So whole genome sequencing, we've gone from in the 2000s, taking 13 years. 2005, we had Watson and Venter having their little competition, and that took about 3 months, to now being able to do a whole genome sequence in 3-4 days, and the costs coming down rapidly. So alongside this work in the NHS, there was the 100,000 Genomes project ongoing. So this was using whole genome sequencing for clinical diagnostics and gene discovery in exactly the same way that we were doing in hematology for bleeding and platelet disorder patients. They were doing that for all different types of patients in the NHS. So this 100,000 Genomes project in the UK was really an exemplar study to show how whole genome sequencing could be used in the NHS, in a healthcare system, to provide diagnostics. And I'll tell you in a minute, I mean, it's been hugely successful, and now we're routinely applying this in the clinic. So of course, I was then at 2017, and you can see, maybe you can't see, so I've got my t-shirt on with my European flag, trying to stay within Europe. I was at the University of Cambridge. I've done a lot of work now on rare disease, thinking about how we can translate that into clinical care. And also I had this huge background around common diseases. And there was really an opportunity, and I really felt very strongly that I wanted to move into into healthcare, into working in the NHS. And this isn't as easy as it sounds. So, a really reasonably well experienced postdoc moving to a whole different way of working. So that in the UK, and I haven't had time to figure out how this happens in Portugal, but I'm sure that I can find out if somebody needs some help with this. So in the UK, what happens is if you want to be a clinical scientist in the NHS from when you leave school and your degrees, then you can do professional qualifications as you go in biomedical or clinical science. But of course, I hadn't done that. So I had to go through what we call an equivalence process. And what this process is.
Kate, just to let you know, you have approximately 5 minutes.
Okay, great. Thank you.
Okay, thank you.
So this equivalence process means that I had to pull together this huge document of evidence to tell one of the professional bodies how much experience I had in different areas. And then I had to have an interview, and they said, okay, fine, you've shown us that you have enough expertise, and we're going to let you register with the Healthcare Professionals Council to become a clinical scientist. So that's really different. A lot of you guys are doing your PhDs, which are academic qualifications, and actually to be a clinical scientist in our healthcare system, we need to have professional qualifications and professional registration, in the same way that a doctor or a nurse has professional qualifications. So in the NHS, I joined in 2019 as a clinical scientist, and very quickly in 2020, things had already started to change, but we had the introduction of the Genomic Medicine Service in England. And so this is where we split the country into 7 different regions. And my region is this turquoise region at this side. So we have about 10 million people that we serve in this region. We're called the East Genomic Laboratory Hub. And I'm now the lead scientist for all rare and inherited disease for this region. So, so that's a really exciting role. I didn't expect to move into the NHS and then move up so quickly. But I think because I have a very varied background, I think the people who interviewed me could see that not only I had the experience for now, I had some really relevant experience for the future. And I'll tell you about that in just a second. So currently now, I think we started our talks to, we could do cystic fibrosis testing and a few other bits and bobs. So now the East Genomic Laboratory Hub, starting from the 1st of February, so can offer, we've got 358 clinical indications that we can test for. And we've got 531 different genetic tests in our scope of accreditation. So this is a directory of the tests that we can do in our lab. So there's a huge amount of stuff that we can now test for to provide treatment options for patients, to provide sometimes just to be able to allow a patient to know what's causing their disease is enough. But also we provide services to allow parents to test whether or not their unborn child has got a disease, so they can prepare for the birth or make other arrangements. And we're really being able to provide clinically actionable results where doctors and healthcare professionals are able to treat patients based on the results that we give them. So this is where I think we're going to go in the future. So this is the next decade, and I think it's going to be more of the same. So the application of the translational research into routine healthcare. And I think that's across the board. That's not just in the UK in the NHS. I think that's in Portugal, that's, throughout the world. The science that you guys are doing in your research institutes, a lot of that is based around human health. And the application of this science is really important, and it's why we do what we do. So I think in the next 10 years, we're going to start to be able to do a lot more pharmacogenomics. So testing people for genetic variants that can indicate whether they can better metabolize specific drugs, or whether or not a drug gives them a risk of an adverse effect. I think very quickly in the NHS and in other places, we're going to be able to start predicting whether or not people are likely to have cardiovascular disease, and then put them on statins early. So I think cardiovascular disease is one of our first things that we'll start to look at with polygenic scores. But I think we will, we'll quickly move on to different disorders where polygenic scores can be really helpful. And I think also at the same time as thinking about the actual science that needs translating, we really have to think about how this happens. So electronic health records and joining together bits of data is really tough in a healthcare environment. And we need to be able to put the systems in place to do that. And then finally on this slide, a lot of this is about education. So not education to people like you and me, education to our doctors and nurses about genetics coming through, and also education to the general public. So we need to have these big conversations about ethics, about sharing data, about making sure that we can test people's genetics and share that data across the world, because those results might help somebody else get a diagnosis. So a lot of this is about thinking about how we educate ourselves and the public and society around some of the really tough questions, and especially for genetics. So that's it. I'm done. Thank you very much. I hope you enjoyed that.
Thank you very much, Kate. Thank you for this excellent presentation, very clear and very interesting. I have lots of questions, but I'll give the stage to the students, of course. If you have questions, please raise your hand. And I'll try to find you out in my screen. Joanna, help me if you see someone, because I'm not seeing anyone. Okay.
I see
João
Vicente.
Yes, João, please.
Not a student, though. Well, we're always studying anyway.
Everybody's a student.
Yes. Hi, Kate. Thank you very much for the talk. I'm curious about something. How is the conversation or the dialogue between clinicians and researchers in the UK? Because here it's not always peaceful, let's say, or not as, probably not as, we don't see each other at the same level sometimes.
Yeah, I think it's similar here. I think some of that comes from the fact that clinicians haven't been educated around genomics, and while they're very expert at everything else, they don't know a lot of genomics. Yes. So I'm gonna say that. I think it is difficult, and I think some of that's around building networks of people who have multidisciplinary skills to bring together some of this really great research to move forward. When you're working at this level with these large projects, clinicians can't do this on their own. They're not statisticians. They don't know how to do cell function work. And I think some communities are better than others. So I know that in the bleeding and hematology world, we have really, really great community internationally. But I also know of other examples where the communities aren't so great. Yeah.
Okay. Thank you. Actually, in my previous work on inborn errors of metabolism, I realized that the establishment of these European networks combining researchers and clinicians, or basic researchers and clinicians, really pushed forward both the treatment and the science for each specific disease. So that would be nice to implement for everything else, right?
Yeah.
Okay. Thank you very much.
Any other question? We have one question from Renata.
Yes, hello, Kate.
Thank
you for a wonderful talk. I just wanted to ask you, on the Human Genome Project, how did you see the competition between the private and the public teams? I mean, how did that happen? And what did you think we won by having that competition? And did we lose something? And if so, what was it?
I think the issue at that point, so that was 2007, and I think the issue at that point was that this Craig Venter's pharmaceutical company was really trying to buy up all the sequencing technologies that were available in the world, and if they'd have won, you know, if they'd have gone ahead and done that sequencing and really not allowed the public effort to go ahead, then what I think it would have meant was that all of those technologies would have stayed behind those closed doors in the pharmaceutical world. So by the funding agency supporting that public project and making that data publicly available straight away, that meant that people like Craig Venter couldn't start to patent genes as he'd started to do. So some people really had started to say, I want to put a patent on a gene. But actually the public genome project was just putting the data out there. And so, you know, basically breaking patent laws, but nobody knew really how to deal with that kind of stuff. But also, I think that just the fact that was done in such a public way, sharing data around the community, meant that it really made sure that the projects that came after it could be done in a more community-based, in a public way. Is that okay? I know that doesn't 100% answer.
No, it is. Thank you.
So yes, I think, for those who are not familiar, the run for the human genome, the enterprise that was the public enterprise versus the Craig Venter enterprise, it's really a fascinating history. That's there are lots of things about it that even involved meetings with the American president. Mm-hmm. And discussing how to publish peer-to-peer science and nature on the same week and so on. It's really a fascinating history. I think we are running out of time, so I will allow one more question from Fredilson. I will ask those people that are on the chat and Tiago Vaz that are waiting, if you could then please contact Kate directly, just because we need to be on time for the next speaker. So Fredilson, please, quick question.
Hi, Kate.
Hi.
Thank you very much for your talk. It was very interesting. So I would like to complement the question asked by João Vicente. So I know that you have just become a clinical scientist, but so far, and maybe with relation to your past experience, what do you think are the dissimilarities between academia and health service? And do you have any hints or tips on how to bring the two institutions close together?
Yeah, so the similarities are, there's a lot of project, for me, there's a lot of project management. There's a lot of doing stuff and getting your sleeves rolled up and getting stuck in and communicating. And I think that works to my skills that I already had. I think what you don't know if you haven't worked in a clinical environment is anything about the quality and the accreditation of places. And so, all the time, I think a lot of our scientists must be looking and thinking about variation and making sure that their experiments are the same every time they do it. And of course, that's absolutely great science. When you have to document that and prove that to an outside assessor, that gets really, really tough. So I think some of the differences are really quite obvious with working in the clinic and academia. I do think that the more and more we bring in new technologies, and the more and more our academic partners work with us together to do research, means that actually when they're setting up translational studies in academia, then we can do a lot of that work with them to make sure that once they have great results, we can bring that straight into the clinic. So I think some of this is about showing that this is possible, and that people as clinical scientists like me want to work with academia and still do work with academia to bring exciting science straight into our healthcare systems.
Thank you.
Okay, thank you very much. The first part of your answer, I would just like to relate it to one challenge that many of you here at ITQB, many of the people are listening. We are conducting some COVID tests for the community.
Okay.
And for the first time, we have over 50 volunteers, many of those are here. And it was really a big challenge for us as scientists to become a diagnostic lab.
Yeah.
And all those that are involved know we have now protocols and quality control at a level that we have never thought possible or even thought about before. And so it relates very well to what you had just said.
It's a really important lesson. And I think you can take some of that back to your bench research. I really do think it's important.
Yes. So we are learning as well through this process. So Kate, thank you very much. Unfortunately, we ran out of time. It was really an excellent talk. Thank you.